Okay, so welcome to Chapter 17 on adaptive immunity. So as a review, there are two main aspects to the immune system. The innate immunity, and that's what we went over before in our last lecture. And that is referring to all of the non-specific defenses against diseases. And the other aspect is adaptive immunity. That's what our lecture today is going to be focusing on. And adaptive immunity is the specific immune defenses against a specific pathogen. There's two main components of adaptive immunity. There's humoral immunity and cellular immunity. Humoral immunity is mediated by B cells, and this involves the production of antibodies against specific antigens. And these are only going to be effective against extracellular pathogens. So the antibodies will be in the fluids, the extracellular fluids of the body, and that is going to bind to the microbes that are on the outside of the cells. The other component is cellular immunity. This is mediated by T cells. And T cells work to kill infected cells. So this is going to be affected, or this is going to affect cells that are already infected. So these are pathogens that are inside of the cells. And this requires the T cells to be able to recognize cells that are infected versus cells that are healthy. And then the two parts are connected. So the innate immunity that we talked about before, as well as adaptive immunity, they have to work together in order to provide immunity. So an antigen is anything that causes the body to produce antibodies. So this can be anything from a protein to glycogen or sugars or anything that's going to cause an antibody to bind to it. Most commonly, these are found on the surface of microbes, and it can also be found on toxins or anything that is ideally not part of the cell. So anything that's an invading foreign body can be an antigen. An antibody is something that's produced by the body. And these are proteins that bind to the antigens, and they signal that this antigen is not part of the cell. So the antibody is, again, made by the body to bind to and signal that this antigen is not part of self and it needs to be taken care of. So antibody structure, all antibodies have this Y-shaped structure. They all have 4 chains. 2 of those chains are heavy chains. These are longer and heavier. And then 2 of the chains are what we call light chains. They are shorter and lighter, and they're on the outside. Here they're in blue. Heavy chains here are in green. All 4 chains are connected by these disulfide bonds. All of the chains have 2 regions. There's the constant region here and the variable region. The constant region is going to be constant for all antibodies. The variable region is the region that is going to change for all antibodies so that they will bind to a very specific antigen. You notice that all of the variable regions are on the ends here. And we can have 2 antigens bind to each antibody. So these 2 variable regions will be specific to one antigen. And over here, we have another antigen binding site. So what exactly do antibodies do? Well, they have several different functions. We have already seen a couple of them. We've already seen that antibodies can help to activate complement. And this was during the classical pathway that we learned last time. We also saw opsonization. So we saw where the antibodies will bind to the surface of a microbe, and that will signal to the to the phagocytic cells to phagocytize that microbe. Antibodies can also perform agglutination, and so they will bind, since we have two different binding sites. We see here they can bind to a couple of different microbes, and we can see more antibodies bind over here, and it can create a big chain or a big blob of microbes. It's basically a way to gather up microbes into a region that's easier for phagocytic cells to take care of, and that's called agglutination. So again, it's gathering these microbes together. They can also neutralize. So if they bind to a microbe, they can prevent that microbe from being able to adhere or enter into the host cells. They can also neutralize toxins. So by antibody binding to a toxin, it will force the toxin to not be effective. And lastly, we see that antibodies have a role in antibody-dependent cell-mediated cytotoxicity. And what this is happening here is we have that is for parasites. Parasites are larger than can be phagocytized. And so what happens here is we have the antibodies will bind to the surface of that parasite, and then those antibodies will signal to immune cells to release substances onto that parasite to kill it. So there's 5 different types of antibodies. IgG. IgG is the most abundant, and it is a monomer. Monomer just means that it's one of those Y-shaped antibodies by itself. We see IgG in complement, enhancing phagocytosis, neutralizing toxins and viruses. And IgG is a little bit special because it can actually cross across the placenta, and it will protect the fetus. And we will also see, since it's crossed across the placenta, we see that in the newborn baby, and it will help to protect the newborn baby for a short period of time. Another type of antibody is IgM. IgM is a pentamer. That means that there's 5 of these Y-shaped antibodies connected together. This is especially helpful in agglutination because now we have 10 antigen binding sites, 2 for each of those antibodies. And so since there's 10 of these antigen binding sites, it can actually bind to 10 different antigens and really help to group all of these microbes together. IgA is a dimer. So we see 2 different antibodies connected by the secretory component. And this is found in mucous membranes, saliva, tears, and breast milk. Then IgD is another monomer, and where we see IgD is on the surface of B cells. So this is actually a B cell receptor here. And lastly, IgE. IgE is another monomer, and it is on mast cells, basophils, and it can also be found in the serum. And where this is important is if a IgE antibody that's on the surface of these mast cells comes into contact with antigen, antigen would bind here, then that is going to cause the mast cell to produce histamine. And we are familiar with histamine in the allergic response. So the antigen and the antibody are like a lock and key. They have to fit exactly for each other. So how do the B cells produce antibodies that are the exact right kind of shape for the antigen that is infecting the body at that time? So to understand that, we have to talk about major histocompatibility complexes, or MHC, much shorter. MHC are molecules that are encoded by the cell to be presented on the cell surface. There's two types, class 1 and class 2. Class 1 MHC molecules are found on all of the cell types in the body. And they present the self of the cell. So it's saying that I am part of the body, this is me, I am healthy. And so that's what we normally see presented on class 1 MHC molecules. Class 2 MHC molecules are instead only found on the antigen-presenting cells. Right now we're talking about B cells, so we're going to stick with B cells here. So inactive B cells will have antibodies on them. And there's lots of different types of B cells with different types of antibodies on them. And these are made randomly. And so when an antigen is exposed to the body, there's a whole bunch of these random antibodies on different types of B cells. Now, what happens to be the right B cell will bind to the right antigen. That's going to help to add that specific B cell. So that B cell will come in contact with the correct antigen. It will internalize it, and then it will present it onto its MHC class 2 molecule. So it's like saying, this is the bad guy, I found it, and we need to work together to kill this. And so what's going to happen is once a B cell has come into contact with the correct antigen, it is going to do what we call proliferate. It's going to divide and multiply itself. And so all of these cells will be exact clones to the correct type of B cell that has the correct type of antibody for that specific antigen. So that's how the body will suddenly produce a lot of antibodies for that specific antigen. So here we have clones of the correct specific B cell, and they will turn into plasma cells. And remember, plasma cells were cells that produce antibodies. So now we have lots of specific antibodies for that specific antigen. So this slide is a little bit complex, I know, but it's also a very important slide. So let's jump right into it. So the way the immune system is activated in the beginning is through antigen-presenting cells. Antigen-presenting cells are going to include macrophages and dendritic cells, are our two main types of antigen-presenting cells. And what that means, antigen-presenting means that they're going to present antigen on their class 2 MHC molecules. And so how they do this is they will actually phagocytize the microbe, internalize it, and then present that antigen on their class 2 MHC molecule. So they're presenting again. This is the bad guy. They're presenting it. Then we also have these cells, helper T cells. And so when the antigen presenting cell comes into contact with the helper T cell, that's going to activate the helper T cell. This activated T cell can now go, this is the activated helper T cell, can now go and activate the B cell. So the B cell has already come into contact with the right antigen and is already presenting that on its MHC class 2 molecule. Then the activated helper T cell comes into contact with that MHC class 2 molecule. And they're in agreement. They both agree this is the bad guy. So we're going to go ahead and activate this B cell. So the helper T cell will release cytokines, which will activate the B cell. And then the B cell will produce all of these clones by proliferation, and they will turn into plasma cells, and plasma cells produce antibodies. Also, B cells turn into plasma cells. Most of them will do that, but some of them will also turn into these memory B cells. Memory B cells will last a long time, if not a lifetime, and so they are ready in case they are ever exposed to that same antigen again. So just to review this slide one more time. Starts with the antigen-presenting cell, ingests this antigen, presents it on its class II MHC molecule. That activates helper T cell. Helper T cell binds to the B cell, which has already presented the same antigen on its MHC class II molecule. Now they agree, activated helper T cell activates the B cell proliferates, produces antibodies. This is the same thing, but I just wanted to point out that this is what we call T-dependent antigens. So most infections are going to require that the B cells be activated by the helper T cell. That's why we call it T-dependent. It's dependent upon the activation by this helper T cell. Now we can also see in some cases where the B cells can be activated independent of the T cells, independent of the helper T cells. This is only when they come into contact with bacterial capsules or the lipopolysaccharides. And in this case, they'll be activated without the helper T cells, but it is a much weaker response, and it's not going to produce any memory cells. Okay, so that's B cells. Moving on to T cells here. T cells are activated in the same way. So they're going to be activated by these antigen-presenting cells and also activated by the helper T cells. If you see CD4, CD4 is the same thing as helper T cell. If you ever see CD8, that's the same thing as a cytotoxic T cell. But anyway, it's activated in the same way. It will come into contact with the antigen-presenting cell, and the antigen-presenting cell will also produce co-stimulatory factors, and that will activate the T cell. A little bit about these antigen-presenting cells. Again, they are mostly dendritic cells or macrophages. Dendritic cells, of course, engulf and degrade these microbes and present them on their MHC class II molecules. And remember that these are the ones that are localized to certain areas. Macrophages do the same thing. They can be activated by cytokines or as well as the ingestion of the antigenic material. But these guys are going to migrate to the lymph tissue to present antigen to T cells. Now there's two main types of T cells. CD4 or helper T cells. We've already talked about these. Remember, they're activated by the antigen presenting cells, specifically activated by the MHC class II on those antigen presenting cells. And they are going to help to activate the T cells, cytotoxic T cells, and the B cells. The other type are cytotoxic T cells. And these are the ones that are going to kill any infected cells. So again, they are activated the same way. Okay, so let's assume that have already been activated. Then they will go to infected cells. Now to understand how this happens, we have to go back to MHC class I. So remember, MHC class I were presented on all cells. And they are showing that they are part of the self. So normally MHC class I molecules are saying, this is me, I'm healthy, don't kill me. But if there is an antigen, there is a pathogen inside of the cell, then it will break down that antigen and present it on its MHC class 1 molecule instead of presenting it. The cytotoxic T cell will recognize that. So it will come and recognize that this cell is infected, and then it will kill it. See helper T cells. So this helper T cell is activated. It will activate B cells. Again, remember B cells are producing these antibodies. Helper T cells can also help to activate cytotoxic T cells. And again, remember cytotoxic T cells attack infected cells. Now, CD8 cells, or cytotoxic T cells, they are already activated in the same way. And once they're activated, they are going to recognize that the MHC class 1 molecule has antigen. Once it binds to that, then it will kill the cell by releasing perforin and granzymes. Perforin helps to puncture holes into the cell, produces these channels in the cell membrane. Granzymes are what's going to initiate apoptosis of that cell. And again, remember, apoptosis is programmed cell death. So now that cell is going to die. To help to prevent that virus generally, but it can be any kind of intracellular microbe, it's going to prevent that from spreading to other cells. All right, and then natural killer cells is the last cell we're going to talk about. Natural killer cells can have roles against virus-infected cells, against tumor cells, and parasites. So the big thing that we see against parasites is we call this antibody-dependent cell-mediated cytotoxicity. That's the process that parasites are killed. So again, a parasite is too big to be able to phagocytose, which is what we do for most microbes. So instead, what's going to happen is they're going to produce antibodies that will bind to the surface of the parasite. Then these immune cells, which can be macrophages or eosinophils or the natural killer cells like we're talking about right now, they will come and bind to the other side, the constant region of that antibody. And that will initiate these immune cells to release the same things. The perforin, perforin's going to kill it. Granzymes will also kill it. And other lytic enzymes will help to digest it. So that was how natural killer cells will kill parasites. And we said natural killer cells will also kill virally infected cells. So very early on, during a viral infection of a cell, there will actually not be any MHC molecule found on the surface of that cell. So that's what we're seeing down here. Again, this is only very early on during viral infection. The natural killer cell will recognize that there is no MHC molecule on this cell. And that will target or trigger the natural killer cell to kill this cell. And it's going to do that in the same way that cytotoxic T cells kill, with perforin and granzymes. All right, so memory. After an infection or also after vaccination, the body will build up immunity. And this is done through those memory B cells. So the memory B cells, again, will live a long time, and they will produce antibody. They won't necessarily produce a massive amount of antibody all the time, but they will be there with antibody on the surface of the B cell. If they come into contact with that same antigen again, then they are already ready and prepared to produce massive amounts of antibody. And so this allows the immune response to be much faster, more intense, quicker or sooner on, and longer lasting. And so this actually will oftentimes be able to overcome the pathogen before symptoms are even seen. Or if symptoms are seen during the secondary exposure, they're going to be much more reduced than they were the first time. Really quick, the amount of antibody in the serum, we call that antibody titer. So what we can see during the secondary exposure, we're going to have a much larger antibody titer much earlier. So we see that here. We see this antibody during the second exposure is much higher and much faster than it was during the primary exposure. So there's several different types of adaptive immunity. Firstly, it's divided into naturally acquired and artificially acquired. Naturally acquired is acquired through natural processes. So the way that the body normally gets infected is naturally acquired. And artificially acquired is acquired through medical processes, so vaccination or injection. It's artificially acquired. And then we have active versus passive. So active is where the body itself is producing antibodies, versus passive is where the antibodies are given to the body. So to go through all four different types of adaptive immunity, naturally acquired active immunity is where antigens are exposed to the body through normal processes, the normal way that we become exposed to pathogens, and then the body will produce antibodies against it. So this is what we normally see. Then during naturally acquired passive immunity, this is of course again natural, but the antibodies are produced, or they're actually given to the body. So this occurs when the fetus receives antibody from the mother. The fetus didn't actively produce those antibodies on its own. It received those, but it's naturally acquired from the mother. Antibodies can also be passed through mother's milk. Artificially acquired active immunity is where we see vaccines. So it's artificially acquired, of course, through the injection of the vaccine. And it's active, though, because once you receive a vaccine, it is your body that is producing the antibodies against that antigen. And then lastly, we see artificially acquired passive immunity. This is where we inject antibodies directly into the patient. So we see that for certain diseases as well as toxins, we can inject the antibodies against that toxin. And the last thing we're going to go over is the process of ELISA. So ELISA is where we use antibodies to be able to identify what type of disease or microbe a patient has. So the first step is we're going to attach antibody into the bottom of this well here. Then the patient sample is added. So if there is this antibody here is going to be specific for a certain type of antigen, a certain type of disease, pathogen, okay? If that pathogen is in that patient's sample, it will bind to the antibody. Then in between each step, everything is washed away. So if it's not bound, it'll be washed away. Then we add more antibody, but this antibody has an enzyme on the other side. And then lastly, there's the substrate that's added. And in the substrate, there are reaction products that are catalyzed by this enzyme. So if the enzyme is there, then these substrates will change color. If the enzyme is not there, then the substrates will not change color. The reason that the enzyme would not be there is if there was no antigen, then none of these other things will stick and they'll all be washed away, and so we won't see any enzyme there. The only way the enzyme will be there is if antigen bound to antibody, bound to the next antibody with the enzyme. and then it will be there. And we will see the color change. Now, usually what you'll see is we can have like 64 well plates or a lot of different wells with a different type of antibody in each well. And then you can put the patient's sample into all of these wells and do all of this process once. Because we have 64 different type of antibodies, we can perform 64 different tests for a patient's sample. So you'll be able to test for 64 different types of diseases. So that is Elisa, and that will conclude our lecture on Chapter 17.